Welcome to Easy Microbiology. Welcome, everyone. Today we will be discussing Listeria seeligeri, a gram-positive, rod-shaped bacterium found in soil, water, and sewage. We'll look at the bacteria's morphology, cultural characteristics, biochemical characteristics, host range, virulence factors, and mechanisms of antibiotic resistance. Let's get started.
Listeria seeligeri is a gram-positive, rod-shaped bacterium that forms part of the Listeria genus. It is non-spore forming and has been identified as the cause of foodborne illnesses in humans. It has also been found to be an opportunistic pathogen, capable of causing severe infections in immunocompromised individuals and pregnant women. Listeria seeligeri has been linked to food poisoning outbreaks due to its ability to contaminate food products, including dairy products, eggs, seafood, and processed meats. The bacterium is capable of surviving in harsh environments and can grow in temperatures ranging from 4 degrees Celsius to 45 degrees Celsius, and pH levels as low as 4.0. Therefore, it is important to pay attention to proper food handling to reduce the risk of contamination and foodborne illnesses.
Listeria seeligeri is commonly found in soil, water, and sewage. It can also be found in some foods, including dairy products and meat.
Morphology of Listeria seeligeri is typically that of small rods, commonly referred to as coccobacilli, with a length of between 0.5 to 0.8 microns and a width of 0.5 to 0.6 microns.
Listeria seeligeri grows well on standard laboratory media, including blood agar, McConkey agar, and tryptic soy agar. It grows slowly and forms small, gray-white colonies.
Listeria seeligeri is a species of gram-positive bacteria exhibiting biochemical characteristics that make it a viable choice for a variety of applications. It is catalase positive, oxidase negative, and has the capacity to hydrolyze esculin. Moreover, this species is able to grow in temperatures ranging from 0 degrees Celsius to 45 degrees Celsius, with the best rate of development occurring from 30 degrees Celsius to 37 degrees Celsius.
Listeria seeligeri can infect a range of animals, including humans, pigs, cows, and chickens.
Listeria seeligeri is a bacterium with various virulence factors, although not all are fully understood. One of these is Listeriolysin O, a toxin that creates pores, allowing it to escape from phagosomes and spread within the cytoplasm of host cells. This gives the bacteria an advantage in colonizing within the host. Strain of Listeria seeligeri has been studied for its ability to resist certain antibiotic treatments. It has been shown that this strain of bacteria can survive ampicillin, tetracycline, and chloramphenicol, among other antibiotics. This demonstrates antibiotic resistance can occur in nature, stressing the significance of staying alert to protect against antimicrobial resistance. Epidemiology tells us that Listeria seeligeri is a rare cause of human infection, with few reported cases. This organism is classified as an opportunistic pathogen and is mostly associated with immunocompromised individuals. This means that it generally affects individuals with weakened immune systems. Pathogenesis investigates how diseases emerge and develop. One such pathogen is Listeria seeligeri, whose systemic infection can take the form of sepsis or meningitis. This bacterium can penetrate and reproduce inside the host cells, hence highly hazardous. Our research has uncovered that the immune response to Listeria seeligeri infection is yet to be understood. To tackle this, we are dedicated to learning and understanding this response, in order to find a successful way to reduce the spread of the disease. We are optimistic in our development of inventive solutions and a sustained focus on sustainability, believing that it will lead to an improvement in public health. To diagnose a Listeria seeligeri infection, polymerase chain reaction, PCR, should be used as it is the recommended technique. This molecular analysis is essential to differentiate between various species of Listeria and correctly identify the strain, thus guaranteeing an accurate diagnosis and treatment. Prevention of Listeria seeligeri infection is paramount for food safety. Proper food handling and cooking, avoiding sources of contamination such as water and soil, and testing food regularly can help to lower the potential risk of exposure. Awareness of the different characteristics and risks associated with Listeria seeligeri can help to detect it quickly, thereby reducing the risk of infection. We understand the importance of proper treatment of Listeria seeligeri infection at our company and offer a range of antibiotics, such as ampicillin, to effectively combat the infection. Furthermore, we provide supportive care to ensure a well-rounded treatment. This slide shows a close-up of unopened pill packets to ensure that our products are safe and effective. It is important to take steps to ensure the food you consume is properly handled and cooked in order to protect yourself and your family from Listeria seeligeri, a rare but potentially dangerous form of foodborne illness. Furthermore, contamination of water and soil should be avoided. Knowledge of the virulence factors and antibiotic resistance of this bacterium is essential for a successful combat against it. Thank you for your attention.